Okay, today we're going to talk about analyzing DNA fingerprints. We have just run the gels through the gel electrophoresis, and we used 4 different DNA fragments to see what's going on. We used one that was an uncut lambda DNA digest, which we would expect only one band from. We used 3 other of the same type of DNA, the lambda DNA, that were cut with 3 different restriction enzymes. One was the PSTL, one was the EcoR1, and one was the Hindi3 digest. And so now what we're going to do is we are going to analyze the DNA fingerprint so that we have some workable data. All right, so what do? Well, we're going to determine the lengths of the fragments, and we're going to do this based on measuring the distance the DNA traveled through the gels. We also are going to plot that onto something called a semi-log graph, and then from there we should be able to estimate the size of the DNA fragments. We use this to mathematically analyze the bands so that we have some quantitative data to back up just what we're observing with the banding patterns. And we also use this information to help create chromosome maps, which we will get to in the next couple of days. Okay, so we should end up with a DNA gel that looks something like this. What you will notice here on the side is something that is referred to as a DNA ladder, and also called the control. And what we use this ladder for, or a standard also, and what we use this for is that this is a DNA sample that has been run, and we know that whatever the enzyme is that we are cutting that DNA fragment with will result in all of these bands, and we actually know the length of each and every single one of these bands and how long they should be, so that we can now use our known fragment lengths to estimate the size of each of the other types of fragment lengths that we have. Okay? All right, so we're going to walk through this process. The first thing that you do is you're going to take the gels that we ran in our electrophoresis machines, and you are going to measure the distance that each and every single one of these bands traveled from the well. And it's important that you make sure that you start at the same location within the well. The easiest thing to do would be either to start at the top of the well right here, and you are going to always measure to the middle of your first band. Okay. Then you come back, you start at the beginning or at the top of this well, and you're going to measure to the beginning of this, or sorry, to the middle of this second band. Okay, and you're going to continue on in that fashion. The other option then would be to start at the base of the well, and then again measure always to the middle of each and every band. And so that way that you're consistent in your measuring. So either start at the top of the band, or at, the top of the well, or at the bottom of the well, but always measuring to the middle of each of the bands. Okay, so this is the first band, again to the second, to the third, and you're going to continue on in that fashion, recording your data in your data table as to the distance that each of the bands traveled. Okay, then you will move over to the next well, and you will continue on in that fashion as well. Again, same location, either from the top of the well or the bottom of the well to the middle of each of the bands. Okay, recording that information in your data table. All right, so then once our turn is time, we're going to go through and we're going to now graph our information. All right, we're going to use something that is referred to as a semi-log graph. It's semi because only half of it is a logarithmic graph and the rest of it is a standard graph that we are used to. The standard graph goes along the x-axis down here. On our information, we are going to be graphing in millimeters. This is just a sample, and this sample graph here shows it in centimeters, but our data will be in millimeters. And so again, making sure that your data or your distances are consistent. So if you start from 0 to the first line is 2, the next one should be 4 millimeters or centimeters, excuse me, on this graph, then 6, then 8. Okay. Now, if we take a look at the y-axis on our data is already labeled, and you will notice that it's kind of interesting. We're going to start from 100 down here. Our next line then will be 200. Then we're going to count by hundreds all up. 3, 4, 5, then we have 8, 900, we get to 1,000, and that makes sense. But now we're going to switch our scale here at 1,000, and rather than counting by hundreds, where this would be 1,000, this would be 1,100, we now are going to graph by 1,000. So this is 1,000. This is going to be 2,000, 3,000, 4,000, all the way up to 10,000. Once I reach 10,000, we now are going to count by 10,000. So 10,000, 20, 30, 40,000 on up to 100,000. And it will continue on that line till we reach the top of the graph. So what we're going to do is we're going to take the data that we got, the distances measured from our standard, from our known fragment lengths, and in our lab that happens to be the DNA that was digested with the Hindi 3 digest. Okay? And that was probably your last well that we had. All right, again, this is sample data here. So on our standard, on our ladder, we know that there are base lengths that are 5,000 lengths long, 4,500, 4,000, and on down to 500 being the shortest base pairs. Okay? The MW up here stands for molecular weight, and it makes sense that the longer or the more base pairs that we have, the more that DNA fragment weighs. Okay, so those go hand in hand. On our sample information, we have measured the length that each of the fragments traveled. Again, you can see that the shorter fragments, 500 base pairs long, traveled the farthest distance because they're smaller. They can maneuver through the gels easier and quicker. It traveled 4.65 centimeters. The smallest or the largest fragment at 5,000 base pairs traveled the shortest distance at 1.8 centimeters. Okay, so your data table will look similar to this. All right, so we're going to go through and we're going to graph this information. Okay, we're going to graph this information here on our semi-log graph. So here we start off, we've got our 4.6 millimeters traveled, went the 500 base pairs or whatever that distance was. We're going to continue on up, graphing all of our data from our standard only. Okay. Then what we're going to do is we're going to create a line of best fit, but we are only going to create a line of best fit using the ones that show a linear relationship, so a straight line. Okay. When we start to see this DNA take on these kind of goofy patterns or start to increase into a curve relationship, we are going to omit that piece of information. Okay. Now, because we have this line of best fit right here, we now can go through and it should be in a solid line because we actually have data for this. We can extend this straight line relationship farther up. And I apologize, I don't know how to do that on my computer here. But if you extend your known data into unknown information or estimated data, this line now from this point on becomes a dotted line. And I would put a dotted line on there, but I don't know how. So on your graph paper, you can extend it. Okay. All right, so now that I have my information, sorry about that, we now can estimate the size of each of our other fragments. If I know that a base pair is, if I know that a fragment on my gel from another digest traveled 3 centimeters on this data, what I will do then is I will go up to the top of this where this line intersects, our line of best fit, and then I can go over to my y-axis, and using this point on the y-axis, then I can estimate that my data is probably, well, it's a little more than halfway, so it's about right here. I'd probably say it's about 1,750 base pairs long. And so that would be the number that I would record in length of fragment for that particular section. Okay, so here we go. In this example, we have a fragment that is 3.8 centimeters long. Okay, I can go up to my line, and then I'm going to come over, and I can see that it's probably a little over 900 base pairs long. Okay, or I can round it down to 900. All right, so what your assignment is going to be is, first of all, you are going to take your data that you graphed from your gel. You are going to plot the Hindi 3 digest on the semi-log paper. You are going to use that information then to estimate the length of all of the other fragments that exist. So the ones that were cut with the EcoR1, the ones that were cut with the PSTL digest, and the ones that were cut with the lambda DNA, okay, which should only be 1 bar. The second thing that you're going to do is that I'm going to give you some sample data, and you are going to, this comes from the lab manual, you are going to record the length of each of those fragments and what they would have traveled on professional person's information, and you are going to regraph that information on a separate semi-log graph. You will then use that data to estimate the lengths of each of the fragments based on real information. Okay? Once you have both of those done, then you will go through and you will finish answering all of the questions in the packet. Okay? So I'm going to try and switch screens right now. See if I can do that. Oh, getting closer. All right. And so what you can see here is that you can see that these are the lengths of the fragments that they should have traveled. Okay? So if we were doing this in a professional lab, hopefully we would have seen all of these. I know the samples that we did in class, we had quite a few of them, more than we usually have show up. So that was pretty good. So this is the information. You go ahead and copy this down into your second data table. The first data table should be your information. Please label that as your information. The second data table should be the sample data. And so you will graph that and estimate the base pair lengths based on all of these distances traveled. All right, I hope that helps.